in Bangladesh. And to solve this problem, varietal screening would be a possible way and organic management would be done, as the organically grown strawberry produce sweetest fruit. Concerning this point in view, the aim of the current study was the evaluation of the performance of strawberry germplasm in different organic methods. Materials and methods. The experiment was conducted at Horticulture or Sher-e-Bangla University. Experiment consisted of two factors. One was 3 strawberry germplasm and another was 4 different types of organic matters, including control. And the experiment was conducted by following completely randomized design with 5 applications. All of the organic matters were applied at the rate of 0.5 kg per pot. Here is the sketch of the experiment. Data were collected on different growth, yield, and quality related parameters. The pictorial views of some instruments that were used on the experiment. Collected data were analyzed by using SRC computer package program and SPSS computer program. Results and discussions. To differentiate 3 germplasm that were used on the experiment, 15 different characteristics were observed. Among them, pictorially differentiated some characteristics of the 3 germplasm. Here the leaf variation among the germplasm. Germplasm 1 provided the curly with rough leaf surface. Germplasm 2 had the spreaded and flattened with smooth surface. And the germplasm 3 had the flattened with smooth surface leaf. Peduncle length variation. Germplasm 1 had the longest peduncle, whereas the germplasm 2 had the shortest peduncle. Flower petal area variation. Germplasm 1 had the maximum flower petal area, whereas the germplasm 2 had the minimum flower petal area. The fruit ripening system unusually among the 3 germplasm. Germplasm 1 start ripening from the middle portion of the whole body of fruit. Germplasm 2 start ripening from the basal portion of the fruit, and germplasm 3 start ripening from the upper portion of the fruit. Here the fruit shape inequality of the strawberry germplasm. Germplasm 1 had the long conical shape, germplasm 2 had the oblate, and germplasm 3 had the short wedge shaped fruit. Number of runners. The graph represented that germplasm 2 and strawberry plants treated with vermicompost provided the maximum number of runners at different stage after transplanting. And germplasm 2 in combination with vermicompost provided the maximum number of runners at different stage after transplanting. So what is the reason behind this? As the vermicompost provided the plant growth regulating substances and improved the soil biological function, that may be responsible for the increasing number of runners, and variation among the germplasm may be genetically controlled. Here the graph represented that germplasm 2 and strawberry plants treated with vermicompost provided the maximum number of stolons, and the germplasm 2 in combination with vermicompost provided the maximum number of stolons at different stage after transplanting. As the number of runner production varied among the germplasm, so the number of stolon production also varied among the germplasm. And vermicompost facilitated the proper growth, enhanced nutrient uptake, and increased the assimilation capacity and hormonal activity. And all of these activities may lead to the increases of stolon number. The graph represented that germplasm 2 and strawberry plants treated with vermicompost provided the maximum number of leaves at different stages of transplanting, and the germplasm 2 in combination with vermicompost provided the maximum number of leaves at different stages after transplanting. So what is the reason behind this for providing maximum number of leaves by incorporation of vermicompost? Because it provided the plant nutrient in readily available form, and afterwards on the vermicompost act as an agent of turning vermicompost to soil increases the number of leaves. The graph represented that germplasm 1 and strawberry plant treated with vermicompost provided the maximum number of leaves at different days after transplant. And germplasm 1 in combination with vermicompost provided the maximum leaf area at different days after transplant. Because vermicompost improves the soil physiochemical properties, increases the enzymatic activity, soil porosity, aeration and water holding capacity, boosted up the microbial population diversity and their activity, amplified the plant growth hormone. And on the other hand, it contains higher nutrient and is readily available for the crops. Besides, vermicompost promoting beneficial microbes that enhance soil biodiversity, that increases the plant growth both directly and indirectly. Directly by supplying the plant growth regulating hormones and enzymes, indirectly by controlling plant pathogen, nematodes and other pests. Similar direct and indirect mechanisms were found by Christiana and George. They proposed a mechanism by which vermicompost may directly and indirectly influence the plant growth and development. The graph represented that germplasm 2 and strawberry plant treated with Horticulture provided the maximum plant height. And germplasm 2 in combination with Horticulture provided the tallest plant at different age after transplant. And it may be that Horticulture supports more vegetative growth of the plant. Horticulture contains higher nitrogen and phosphorus, also more water retention capacity that may lead to the plant height increase. From the experiment, it was found that germplasm 1 provided had taken the minimum days for flower bud initiation, flowering, fruit setting and fruit harvesting, whereas vermicompost treated strawberry plant provided had taken the minimum days for flower bud initiation, flowering, fruiting and harvesting. And germplasm 1 in combination with vermicompost provided the best result in terms of this crop duration related activities. As the incorporation of vermicompost caused the early flower bud initiation, flowering, fruiting and harvesting that minimizes the cropping period that may lead to increase the cropping intensity. And early flower bud initiation, flowering, fruiting and harvesting may lead to the better production of strawberry with quality in Bangladesh. On the other hand, low temperature is required for better strawberry production and with better quality. But from the month of February, in Bangladesh, temperature increases rapidly, and this temperature increases cause the dramatically decreases of the strawberry production quality. And to overcome this problem, the increases of temperature during the month of February, early flower bud initiation, flowering, fruiting and their harvesting is essential. That is, short crop duration is important for strawberry cultivation in Bangladesh, as minimum low temperature period during the winter season in our country. From the experiment, it was found that germplasm 1 provided the maximum number of flower, fruit weight and total fruit weight, whereas the vermicompost treated strawberry plant provided the maximum number of flower, fruit weight and total fruit weight. And germplasm 1 in combination with vermicompost provided the best result in terms of these yield related attributes. So what's the reason behind the increasing number of flowers? Because vermicompost contains microorganisms that produce hormones and humans that increases the number of flowers. On the other hand, vermicompost regularly supplies the plant nutrition, plant growth regulators and hormones. And variation among the germplasm may be genetically controlled. The reason for the increasing the number of fruit, because vermicompost provides the greater supply of humic acid, outstanding physiochemical and biological properties, and larger population of bacteria, fungi and actinomycetes. And these characteristics may lead to the more number of fruit production. Reason for the increases the number of fruit weight. Vermicompost enriches in nutrient content that leading to increased uptake of NPK and food, and it also reduces the carbon nitrogen ratio the experiment, it was found that germplasm 1 provided the longest fruit, maximum fruit diameter, and highest trees, whereas strawberry plant treated with germplasm provided the maximum fruit diameter and highest trees. Germplasm 1 in combination with vermicompost provided the best result in terms of these characteristics. So what are the reason for increasing fruit length and diameter? As the germplasm 1 and vermicompost post the maximum behavior that enables enhanced carbohydrate concentration in crown and root and fruiting guide, and these adequate root starts generate new feeder roots to provide carbohydrates for flower bud initiation and fruit development that may lead to increases of fruit length and diameter. For the strawberry production, slightly acidic soil is necessary, and humic acid from the vermicompost enhances root elongation and formation of lateral roots that enhance the nutrient uptake. By increasing the permeability of cell membrane, stimulating root growth, and increasing the proliferation of root hairs. Besides, vermicompost contains all of the essential macro and micronutrients readily available for the crops. And this micronutrient may lead to increases the sweetness of the strawberry. Correlation and regression. From the correlation study, it was found that growth related parameter leaf area is significantly positively correlated with all of the related parameters. Here the regression line between the variables of leaf area and root weight, where the regression value R square is 0.9112, and the regression line between leaf area and leaf percentage, where the R square value is 0.8089. As the vermicompost treated strawberry plant and germplasm 1 provided the utmost leaf area and on the other hand maximum yield with sweetness. That's why it may be stated that there is a positive relationship between leaf area and yield with sweetness. How leaf area related with yield and sweetness? Because largely provided much more total photosynthetic area of leaf blades. Despite of their pure leaves, that provided the thicker petioles and flower stalk, that provides the large fruit, ultimately more productive. On the other hand, largely maintain the source-link balance, had supply of assimilates per unit of crops, and markedly improve the sugar content. Increasing leaf area cause synthesis of assimilates due to the enhanced rate of photosynthesis that increases the rate of translocation of photosynthetic product from the leaves to the developing fruits, and that may be responsible for increasing total yield and sugars. Conclusion. Germplasm 1 and vermicompost was the most excellent in case of yield and sweetness, and leaf area significantly positively correlated with yield and sweetness. After further trial, it may be recommended to use germplasm 1 for the production in farmer feed and vermicompost to improve the sweetness. Further research are suggested that to improve the seedling production and their management, to solve short low temperature period and rapid high temperature during food ripening, and improvement of post harvest quality. Finally, I would like to acknowledge about my research work. I endeavor to Almighty Allah, my sincere gratitude to respected supervisor, co-supervisor, and teachers of the Department of Horticulture. I am very much grateful to my parents and thanking to my classmates.